This study presents a novel workflow to detect SARS-CoV-2 on surfaces that are rarely cleaned in the urban environment, such as gas pump handles, playgrounds, and ATMs. In a pandemic, supplies are scarce. We use easily obtainable materials and reagents, and equipment available in basic laboratory settings. We use an extraction method that preserves the RNA without a cold chain, and a detection method resistant to inhibitors. This protocol is of public health interest. It provides a framework for the assessment of environmental viral reservoirs for the current COVID-19 pandemic and other infectious agents during future global outbreaks.
Recruit citizen scientists using a direct and clear call to action released via local and social media. Create a social media handle to connect the topic across social media content. Create a link to the SMP, providing a multilingual plugin to enable navigation in multiple languages for individuals to apply to participate in the environmental sampling effort by answering biosafety-related questions specified in an online form. Include in the sampling section graphic and audiovisual protocols in English and Spanish. Visualize geospatial data using a geospatial application programming interface facilitated by a cloud computer service provider. Store the data submitted to the LIMS through the SMP to facilitate centralized storage, tracking of processing workflows, and management of the logistics. Preload information such as sample kit ID,